In the last video, I talked primarily about ribosomal RNAs as well as ribosomes. In this video, I'll be talking about transfer RNAs and messenger RNAs and evolution of the genetic code to bring all the translation information together. And I'll also talk about small RNAs that are also involved in some of these processes. So first I'll discuss transfer RNAs, transfer RNA genes, otherwise known as tRNAs. And these are transcribed by RNA polymerase in bacteria and archaea and polymerase 3 in eukaryotes. Here's a typical structure of a tRNA with the acceptor arm up here, which contains the 5' end and the 3' end, to which has been added CCA, and that's added post-transcriptionally. And then here's the D domain here, which contains a dihydroxyuridine, and then an anticodon loop, which has the anticodon down here that attaches to the codon in the mRNA. And then the T domain here, which contains a thymidine, normally found in DNA, but here it's found in RNA. And then back up to the 3' end of the acceptor arm, and that's where the amino acid is attached right there. Here's a three-dimensional structure of the tRNA. So it has this letter L shape. This is the CCA on the end here in the acceptor arm. There's the D domain or D loop there. There's the T domain there, and this is the anticodon loop down here, or anticodon domain with the anticodon right here on these three nucleotides. Here are slightly different views. This one here with the acceptor arm showing some of the conserved nucleotides. In most of the cases, the nucleotides are not conserved, but in this case, there are a few A's conserved here and a pair of G's here. There's the anticodon loop with the three nucleotides in the anticodon, and usually a uridine in that position. A variable loop here, and then a T pseudo-C or T domain, which usually has a thymidine, a pseudouridine, and a cytosine there, preceded by a G in a GC pair. On the D loop, there's often a dihydroxyuridine as well. And this is just a sort of a shadow diagram of what that looks like with the amino acid attached in each case. So here are two genes, the tRNA genes and the 5S rRNA gene. These are both transcribed by RNA polymerase 3 in eukaryotes. And it shows you that part of the promoter, in this case, in both cases, is within the gene, that is, within the expressed part that becomes the mature tRNA. Now, in mRNA genes, the promoter is upstream from the start of the transcript, and in ribosomal DNA, the promoter is within the 5' spacer that gets cut out later. So there's some variability as to where the promoter region is in each case. So within a tRNA gene, there is, within the RNA that's actually expressed, there's what's called an A box and a B box. And transcription factor 3C binds with that region. And then that attracts transcription factor 3B, which then binds there. And that then orients the RNA polymerase 3 to the promoter region to start transcription. In the case of a 5S ribosomal RNA, which is also transcribed by RNA pol 3 in eukaryotes, there's a C box, and in this case there's transcription factor A attaches to the C box. That attracts transcription factor 3C, which attracts transcription factor 3B, which attracts the RNA polymerase to start transcription of that gene. And each one of the transcription factors is actually more complex than that, consisting of several subunits. So here's transcription factor 3C in yellow, and it's the 138 megadalton subunit and the 95 megadalton subunits that attach to the B and the A boxes respectively. And then here's this transcription factor 3b, which actually has a TBP or TATA binding protein. But it doesn't have a TATA box there. So the reason the TATA protein was called the TATA protein is in mRNA. It attaches to a TAA, a TATA site. But here, in this case, it doesn't have a TATA box on the DNA. So that's transcription factor 3b. Up here is pol 3, RNA polymerase 3, which has multiple subunits. That this 128 megadalton subunit sticks to the 95 and 62 megadalton proteins of transcription factor 3C, and then these ones down here, 34, 31, and 82 attached to the 90 megadalton protein of transcription factor 3B. These central ones, the 160 megadalton and 128 megadalton proteins, those are the central parts, and those are in common to all RNA polymerases, including the RNA polymerases of bacteria, archaea, and eukaryotes. Most of the other subunits on here are more specific to eukaryotes, and in fact are more common in the RNA polymerase 3, although some of the subunits also are present in RNA polymerase 1 and RNA polymerase 2 of eukaryotes. So here's a diagram showing some of those evolutionary similarities or connections between the eubacterial RNA polymerase here, having those two main subunits of the RNA polymerase enzyme, as well as some other subunits, and then you add on to those others to get the archaeal types. And then there's this common polymerase down here that eventually diverges into the three different ones you see in eukaryotes, including RNA pol 1, which has these two special subunits down here, and that transcribes ribosomal RNA. And then that's similar to this RNA polymerase 3, but it also has additional subunits, and that's the RNA polymerase 3 that transcribes tRNAs, 5S RNAs, and other small RNAs. And then in the middle here you have RNA polymerase 2, which transcribes only the mRNAs. And here's an artist's rendition of transcription of the tRNAs and 5S ribosomal RNAs. The tRNAs then have this transcription factor 3C that attaches to the A and B boxes of the gene, and then the TBP, the transcription factor 3B that contains the TBP, TATA binding protein, and finally RNA polymerase 3 attaches and starts transcription. For 5S genes, then you need this transcription factor 3A that attaches to the C box, and that then attracts transcription factor 3C, that attracts transcription factor 3B, and finally RNA polymerase 3, which then begins transcription here. Although many tRNA genes do have the promoter, there are some that don't have a promoter any longer, and instead they're transcribed in operons, so that the first gene in line would have those promoter regions to start transcription. But then the other ones beyond that would be transcribed in one long transcript here. So you have, and this is in eukaryotes, have a long transcript. You get processing in the nucleus to about 300 nucleotides each, and then further processing to cut it down to the final 70 to 80 nucleotides, and to add the CCA onto the 3' end so that it's ready to accept the amino acid at that end. Next, I want to discuss mRNA genes, and these are the ones that most people talk about, so I won't go into great detail about these because usually people know more about messenger RNA genes than they know about any other type of gene and RNA type. For many or most bacterial mRNA genes, they are transcribed as an operon containing more than one coding sequence for more than one protein. And so this is the lac operon here, including the lac-I gene and then the lac-Z, Y, and A genes, all that are transcribed as one long piece of RNA, and then they're translated into 3 separate proteins. So that's the operon part. The lac-I gene is transcribed and then translated into a repressor protein. And that repressor has a site, recognizes a site within the promoter region for the lac-Z, Y, and A operon. And so that will attach to that site and form sort of a curled loop, which will not allow the RNA polymerase then to attach to the promoter region, thus preventing expression of that operon and all of the proteins that come from that. However, if you have lactose in the cell, then that also attaches to the repressor protein. If the lactose attaches to the repressor protein, it cannot attach to the operator site. And so it releases from that operator site, and that opens up the region for the RNA polymerase to attach to that site and transcribe that RNA, that operon. When the gene is transcribed, it's transcribed as one long transcript containing messages for 3 separate proteins: the lac-C protein, which is the beta-galactosidase, and then the lac-Y and lac-A proteins that are involved in permeability of the cell to the lactose, so that they can bring the more lactose into the cell. So in this first part in the lac-Z gene, there's a Shine-Dalgarno sequence, which attaches to the anti-Shine-Dalgarno sequence on the small subunit ribosomal RNA. So that attaches it to the small ribosomal subunit. And then the ribosome large subunit attaches to that, starts translating the protein from that, the amino acid sequence, forming the lac-Z protein, ultimately winding up in this stop codon sequence, which is UAA, 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 essentially saying stop translation. Then there is another Shine-Dalgarno sequence after that, that positions that mRNA in the small subunit. There's an AUG start. You make the lac-Y protein, and then there's a stop to stop that translation to make that protein. Next, there's another Shine-Dalgarno sequence to, again, position that mRNA for reading the next part of that message. And then a UUG, in this case, rather than an AUG, this one's used as the start codon, go through the entire sequence to read the amino acid sequence in the ribosome to make the lac-A protein, and finally a stop codon after that. Each of these dashed areas in between that I put in here, that's just the coding sequence for that protein. I left it out because it becomes too cumbersome, and I left in only these parts that are important to the discussion here: the Shine-Dalgarno sequence, the start codon, and then the stop codon. The mRNA genes in eukaryotes differ from those in prokaryotes in that almost all of them that I know of, are individual genes, and you don't have the operons, except in the case of tRNAs. But for mRNA genes, they're all separate genes. They can make more than one protein, so there's another way to make more than one protein, and that's through alternative splicing. So I haven't put that in here, but I have put exons and introns. But you can splice these in different ways to come out in the end with different mRNAs and thus different proteins. So in this example, you often have upstream and sometimes downstream enhancers, which tend to concentrate transcription factors in the promoter region of the gene. And the promoter usually consists of a GC section and a CAT box, and then there's that TATA box, which binds with the TATA binding protein in one of the transcription factors. Then there's a transcription start site, a 5' untranslated region, several exons and introns, and then a 3' untranslated region within that gene. So there's transcription of this portion, starting from the 5' untranslated region, through the exons, introns, and through to the 3' untranslated region. So this is still an immature mRNA. It then, while the transcription is still going on, a 5' cap, which is a 7-methylguanosine, is attached to the end. And then the introns are spliced out one by one, usually in order. And finally, a poly-A tail is added to the end of the mRNA. And now it's shipped from the nucleus and attaches to ribosomes and then is translated into proteins, either on ribosomes that are on the endoplasmic reticulum, the rough endoplasmic reticulum, or within the cytosol, depending on the final destination of those proteins. As I showed that these enhancers attract certain other proteins to them, and those also interact with the transcription factors and RNA polymerase to enhance or increase the degree of transcription of those downstream and upstream genes. There also are things called silencers to which repressors attach, and these will actually tie up that enhancer region so it doesn't stimulate transcription of those particular genes. So these are another way to highly regulate transcription and control of transcription of certain genes. The eukaryotes, eukaryotic mRNA genes, usually have introns, and some have multiple introns. The longest one I know of is the dystrophin gene, that is responsible for, when it's faulty, for causing muscular dystrophy. And I think it has somewhere around 75 or so introns and exons, and the gene is very, very large, over 2 million bases in length. But these are a little smaller. Here's the globin genes containing 3 exons and 2 introns. Here's an ovalbumin gene in bird eggs, which has 8 short exons and 7 introns, and the conalbumin gene that contains 17 short exons and 16 introns. And so you see, as you get more and more introns, the tendency is to have a smaller coding sequence and a larger proportion as introns. And in the case of the dystrophin genes, about 99% of the gene is made up of introns, and only a small portion contains exons. And for the worst form, the most severe form of muscular dystrophy, the problem is just a single nucleotide change that causes a stop codon in one of those exons. The introns still function normally. So here's one example in the case of ovalbumin with its 7 exons and 6 introns, as well as a 3' untranslated region and a 5' untranslated region as well. So here's the gene itself. It gets transcribed into this transcript, and then there's a poly-A tail added to it, and finally splicing. So you start with this primary transcript, which is fairly long, 7,700 nucleotides long, and wind up with a coding sequence in there that is only 1,872 nucleotides in length. So then after splicing of the message, the next step that requires both tRNA and ribosomal RNA is that the message, the RNA, is translated into a polypeptide sequence, which we call proteins. At least above 50 amino acids is called a protein, and below that it's simply called a peptide. So if you look at this genetic code, and this is the quote universal code because there are some changes to the code, so it's not completely universal. But you can see there's some structure to this genetic code. Here's the first position of the codon, second position of the codon, and third position of the codon. So for example, AAC would be A, and then C, and that would encode asparagine to be placed, to be added on to the growing polypeptide chain. And here's the start codon, the AUG for methionine, which is right here. But you notice in this code, so on this side, all these things in this orange color, those are non-polar amino acids. And then everything in purple, those are all polar amino acids. Anything in green, these two acidic amino acids, aspartate and glutamate. And then the basic ones here in light blue, such as lysine. So two questions that always came to my mind were, where does this structure come from? And does that indicate anything about the evolution of the genetic code? It's clear that this genetic code is fairly complex and took quite a few years, probably 1 billion years, to build up to at least this complexity and more. So if you start dissecting this genetic code apart a little bit, you can see that the hydrophobic amino acids have this structure. The first nucleotide can be almost anything. The second one's usually a U, but sometimes a C or a G. And the third codon position, the third nucleotide, is almost anything. For polar amino acids, then the first position is usually a U, but sometimes an A or a C. Second position is usually a C or an A, and then occasionally a G. And the third position is variable. For an acidic amino acid, that is aspartate or aspartic acid, and glutamate or glutamic acid, it's always G, A, and then something for the third position. And the basic ones are either a C or an A in the first spot, an A or a G in the second spot, and the third position is variable. So again, you can see there's a great deal of structure in this genetic code. All the parts of the genetic code and translation in general started much simpler, probably somewhere around 3.5 to 4.1 or 2 billion years ago. And so if you look at the amino acyl tRNA synthetases, which put the amino acids onto the tRNAs, as well as the tRNAs, mRNAs, and ribosomal RNAs, they were a lot simpler in the beginning. So these are some models based on information from the genetic code, as well as information gleaned from studying ribosomes and mRNAs, as well as the amino acyl tRNA synthetases. So to start down here, the ribosomes were fairly simple in the beginning. The small subunit probably just was a carrier of RNAs in general, a delivery system, and the ribosomal RNA was somewhat enzymatic at that time. So it was probably already capable of adding amino acids to a growing polypeptide, but it was fairly rudimentary. But by about 3.6 to 3.8 billion years, it clearly had the peptidyl-transferase in a more modified form, so it was more efficient at adding the amino acids to the growing polypeptide chain. tRNAs appear to have begun just with the CCA, and that's where the amino acids were added in the beginning and where they're still added on the current tRNAs. Then the acceptor arm appeared, and it probably was already somewhat formed by that time. And then the T pseudo C arm was added to that. And these little arrows indicate where introns have been found in those. And it may be that the introns were responsible for piecing these molecules together, both for the tRNA and for the ribosomal RNAs. Then there was a duplication of this whole section, which then gave the 4 different arms or domains of the tRNAs eventually, and then a variable loop to increase the variability of the tRNAs, and finally there are tRNAs that can be sort of spliced together or pieced together in pieces, so you have even more diversity in the tRNAs. During this time also there was further evolution and pieces added to both the small subunit and the large subunit. Again, added, it looks like, in pieces, in larger pieces, and there was further elaboration around that peptidyl transferase site to then add an A site, which was already there, and duplicate that to become the A and the P sites, and then an E site for the exit site, and growing larger and larger around mainly the periphery. So the central part of the ribosomal RNA was the same, the central part of the ribosome, and then you add these layers like an onion to that during each phase to get larger and larger and more discrete, and having a few more functions, but mainly more discrete. When does that ribosome translate and when doesn't it translate? The mRNA seems to have gone through first a small single strand phase, and then a double strand phase for a time. And this relates back to these aminoacyl tRNA synthetases. And that's essentially, there are 2 types, a class 1 and a class 2. And one attaches to one side of the tRNA when it's adding the amino acid, and the other class attaches to the opposite side of the tRNA when it's adding. And they seem to be self-complementary as far as the amino acids and the codes that they recognize. And that's why it looks like originally you could read one message off of one strand of that double-stranded RNA, and then a different message going in the opposite direction, reading from the other side to get a different message and a different protein. But it looks like then the mRNA went back to a single-stranded phase, probably partially because RNA doesn't form very good double helices, so that probably wasn't very stable. And the ribosome was getting larger, so it was probably more difficult to get a double-stranded RNA threaded through a ribosome than a single-stranded molecule. The aminoacyl tRNA synthetases, probably way back here around 4 billion years ago, probably were RNAs. And then eventually, after a few 100 million years, the RNA world shifted to more of a DNA and protein world, and now you were building proteins to perform those same functions. And so it was probably they were more efficient, and as they got larger, they could recognize more and more amino acids and pop those onto a more diverse set of tRNAs as well. So now if we go back to this diagram of the genetic code, and we see the structure of the nonpolar, polar, acidic, and basic groups, and you can sort of exclude glycine because it's pretty much a placeholder. It only has a hydrogen, so it could almost show up anywhere. How did the structure form? And if we had a much simpler translation system, this also must have been much simpler. So how did it start, and how did it get going and develop into the more complex system that we see now? So in the 1950s and later in the 1960s, and then some things that were found in the 70s and later, we know that there were amino acids available on Earth and coming from meteorites also. The Murchison meteorite, and then Stanley Miller did some experiments looking at the ancient atmosphere, and then built some experimental equipment to show what sorts of amino acids can be built without any biological system. So Stanley Miller found these ones, and especially early on, he found things like valine and glycine and alanine in his experimental apparatus after only a week or so of action. So he also later, he and others who worked on this, found all of these amino acids listed in red. So glycine, aspartate, glutamate, alanine, valine, isoleucine, threonine, proline, leucine, and serine. And if you look at their structures, they're fairly simple, starting here with glycine. That's just the R group is only a hydrogen. There's a methyl group here in alanine, and then an ethyl group in valine. So very simple compounds, very simple amino acids. They found other amino acids, but they are not found in biological systems, so just to mention that. Also, the Murchison meteorite, as well as other meteorites that have been found, also have these same 10 amino acids. So these these are among the earliest and simplest amino acids that probably were available even from the beginning of the formation of the Earth, before any biological systems, any organisms evolved. So this points out one thing that biology started, that is, the first organism started with compounds that were already available before any organism appeared on Earth. But still, the genetic code arranged this way is still confusing. Where did things start? You can assume they started from these early compounds and then worked up in this direction, but it's still somewhat unclear. However, you can rearrange the genetic code based on the aminoacyl tRNA synthetases, the AARSs. So all of the white ones up here, all of those amino acids and those triplet codons, those are the ones that are added by the class 1 AARSs that recognize the tRNAs on one side. And then the ones in black are the class 2 AARSs that attach to the opposite side of the tRNAs, and those are the amino acids that they place onto those tRNAs, recognizing the tRNA specifically. And they place the amino acids onto those tRNAs, usually ignoring the anticodon. So many of these aminoacyl tRNA synthetases never touch the anticodon and the anticodon loop, but they're recognizing the upper portions of the tRNA. And these are really what set the genetic code. So these aminoacyl tRNA synthetases set the genetic code, and then the anticodon later determines where to put each of those amino acids based on the mRNA code attaching to the tRNA anticodon. So with this rearrangement, you can start to attempt to, anyway, decipher how the evolution of the genetic code proceeded. And then other evidence comes from studies that have looked at the codons that were present and the amino acids that were present in very early proteins, the most ancient proteins, versus the codons and amino acids present in more modern proteins. So the ancient proteins are those that are in all organisms, right? They've been around from the beginning, so all organisms, all the way back through bacteria, have those proteins. And here on top, those are the frequencies of the amino acids in those proteins. So from this, you see that alanine, valine, glycine, and others were among the first amino acids in the genetic code. They were the ones that you find most often in the genetic code. And the initial nucleotide for each one of those is a G. So that probably was where the genetic code started. And also you notice it would be GC and then GU and GG and GA. So all of the second nucleotides match with one amino acid coming from a single unique nucleotide in the middle position of the triplet code or codon. So that points directly to this bottom group. There's the G in the first position, a U, C, A, G standing for each one of these amino acids on the bottom row, including valine, alanine, either aspartate or glutamate, and glycine, and then anything in that third position. And so that's quite variable in the third position. Then you can make a case for going up in this direction, adding isoleucine, threonine, leucine, proline, serine. And if you look back on that previous slide, here are serine, proline, isoleucine, leucine. All of these are among that group that's also pretty high in frequency in the most ancient proteins. By the time you get to modern proteins, that breaks down. So some of the other amino acids start increasing in frequency, and those original ones start decreasing in frequency. And some of these that are in the lowest frequency are the amino acids that appear in the upper right-hand corner of the genetic code, including cysteine, tyrosine, tryptophan, and others up in this upper right-hand corner. But this has to also be consistent with the frequency of mutations. That is, it's easier to make a single mutation than a double mutation. It's easier to have a transition than a transversion. So this model I came up with a few years ago and published, where here with this gray background, those are the original amino acids and codons that probably first appeared in the genetic code. And that includes glycine, alanine, aspartate or aspartic acid, all which use the class 2 aminoacyl tRNA synthetases, and valine and glutamate that use the class 1 aminoacyl tRNA synthetases. And the codons are listed there, as well as their overall frequency within the ancient proteins. And then down below, the PPOF, those stand for places in biochemical pathways where they can be produced, where those amino acids can be produced or are produced. And so all of those have to be consistent with evolutionary models. And so this goes through valine and then isoleucine, leucine, and finally methionine was a late arrival. And this pathway goes into serine, threonine, proline, and finally histidine and arginine and lysine. And over here, and cysteine is down here, and then you have this one going into asparagine and tyrosine, and down here finally adding tryptophan and phenylalanine. And these ones that appear on the ends of each one of these pathways are some of those that are in the upper right-hand corner of the genetic code, and the ones that are in the lowest frequency. And ones over here are early in the genetic code and in the highest frequency. And each of those changes uses only a single nucleotide change in each case. So this is one way that at least I tried to make sense out of the evolution of the genetic code. So finally, I want to talk about other non-coding RNAs, and these are primarily small non-coding RNAs. tRNA, right, it's a non-coding RNA, and ribosomal RNA is a non-coding RNA. But I want to talk about other types of non-coding RNAs. So here's a diagram of non-coding RNAs. The only coding ones are these ones right here, mRNA. Everything else is non-coding. including ribosomal RNA and tRNA, which we've already talked about. But then there's small nuclear RNAs, including small nuclear organizing RNAs, which process the ribosomal RNAs. And then there are line C RNAs, there's antisense RNAs, silencing RNAs, microRNAs, as well as Pee-wee RNAs and others. So these all kind of harken back to an RNA world, where RNA did most of the processes and proteins did very little. These ones currently are either involved in processing and modification of other RNAs, or they control the expression of mRNAs and other types of RNAs. So if you look in the cell cycle and look at where these are affecting different processes, you have microRNAs affecting alternative splicing, SNO RNAs, which will do nucleotide modification and processing of ribosomal RNAs and other RNAs. There are microRNAs here that cause reduced transcription and translation of mRNAs. And there are other types of RNAs, including some that are silencing RNAs. Human females have 2 X chromosomes. There are RNAs that silence one of the X chromosomes, so you only get one of the X chromosomes actually expressing genes, and the other one is silent. Here's one example of how the RNAs are involved in processing, that is, of the ribosomal RNAs, where the RNA is doing all these processes. The SNO RNAs primarily are attaching to the large transcript, and then they're processing, cutting out certain parts, and also modifying certain bases within those final products. So here in black, that is the ribosomal RNA transcript, starting with the external transcribe spacer here, and then running through the 18S gene and the 5.8S gene, and then the 25S gene, or the small and large subunits. And then here's one of the SNO RNAs, the U3 SNO RNA, that hybridizes to certain parts of that gene, and then starts doing different processes, including cutting right here at that first site, cutting off the 5' end, and then up here forming a loop, and then also clipping that other part off of the primary transcript, and then going into this area, these box A prime, those will begin to modify certain bases. So mainly the uridine bases will be altered in one way or another. And then these boxes also, box C and box D, also can alter bases in RNAs, including those in ribosomal RNA and those in tRNA. That is, you wonder how you get a pseudouridine. Those are done by the SNO RNAs, and how to get a dihydroxyuridine. Those are also accomplished by these modifying RNA molecules. So another set of small RNA molecules comes from a particular genes, which we originally thought weren't really genes. They're small sequences, intergenic sequences. Sometimes these come from introns, so many of them come out of the introns. And then there's a RNA protein complex called dicer that clips these into pieces that are about 21 to 24 nucleotides in length. They then attach to a RISC protein, which is an RNA-induced silencing complex. And then through activation of ATP, it becomes single-stranded, and it attaches to mRNAs. It then silences those. They will not be translated, and they're usually stored somewhere in the cell for later translation or destruction. depending on whether it's a silencing risk or a microRNA risk. And the microRNA risks, again, the microRNA comes usually from introns, and the silencing RNAs usually come from a different transcription, a separate transcription of those. So depending on what type of risk these are incorporated into, these ones that are silencing RNAs in silencing risks will attach to the mRNAs and they'll start chopping them up. So they're essentially silenced. They don't work anymore. On the other hand, if it has a different RNA in it, a microRNA, then it will simply silence or simply store the mRNA without any translation. Sometimes they decay, other times they're stored. If they are stored, they can be released from that complex immediately and immediately go into translation, attached to ribosomes and go into translation. So this avoids having to go through transcription and then processing, export out of the nucleus, and then translation. These are ready to go. So you can have very rapid protein synthesis of these mRNAs that are stored in what are called P bodies. So these are just a couple of categories of RNAs that are in control of both transcription and translation, as well as some that are involved in RNA processing and modification. So then this circles back to this slide I showed in a previous video, that RNAs can be messages in the form of mRNAs, which I discussed today. But they can also be structural, that is, parts of the ribosomal RNAs, parts of the tRNAs, and others. They can be enzymatic. Ribosomal RNA is enzymatic. It has the peptidyl transferase activity on it. And then introns, especially group 1 and group 2, are enzymatic in that they are self-splicing. And finally, there are these controlling RNAs, these small non-coding RNAs that can do anything from process a ribosomal RNA, modified bases, control transcription and translation of certain messages, and can cause degradation of some mRNAs as well. So quite a few different functions of RNA molecules, which also hints to an RNA world that was active long ago, and much of which is still with us in modern cells and organisms. Okay, so that's all for this video. I talked about tRNAs, messenger RNAs, evolution of the genetic code, and then finally these small RNAs, and then tried to tie together these last several videos. Okay, that's it for now. I will talk to you later.